Dear colleagues, I'm happy to see you all, and I'm happy to present our today's speaker, Mikhail Ignatov, who will tell us about Paleozoic mosses and their relationships to extant taxa. Please, Mikhail.
Dear Natalia, everybody, I'm very happy to have such possibility to share my small results to such eminent audience, and it will be as written here comparing of Paleozoic taxa with extant taxa. And extant taxa, I should do some small introduction. They are usually classified by sporophytic characters, especially peristomial. From here you see that peristome for mosses is something like a flower for angiosperms. But unfortunately, the oldest known peristome is Cretaceous, so nothing in Paleozoic. But fortunately, the most early divergent lineage of mosses, Takaki, Sphagnum, and Andrealian mosses, they are peculiar enough in gametophytic character as well. So it is more or less possible to discuss things. And what we will talk about actually. What are material we have at hand for this discussion? Actually, the Paleozoic mosses usually is something yellow on rock surface, but if you solve them in chloric acid and fluoric acid, it become better, and in slides even better. Now I can show you work how it looks. It's not something selected, it's just average material. So you see it is quite nice, and from this we actually can find quite many interesting characters, and of course most of them are leaf characters. So I have to say a few words about leaves, because leaves of mosses are not the same as in vascular plants. They are originated from one cell, as shown here. And the next slide is especially important, because this issue is somehow very difficult for students. It's very difficult to believe that a cell which is split off the apical cell at the end of leaf development appears at the bottom of leaf. Here you can, is my mouth is seen there?
Yeah,
I'm pointing, yeah, so the cell is divided from apical cell, first cell, second cell, third cell, fourth cell, and so they are slowly moving to the basal part of a leaf. And if you look at the apical bud of any moss, you will find some leaves with still undivided cells in the basal part, like here. And this is actually quite important for further discussion. Also, a few words about fossil records in Paleozoic. In Carboniferous, there are very few, and most of them are difficult to link to something extant. So we will not speak much about Carboniferous plants. And the situation is drastically changed in Permian, when quite many places you see here, and in some areas, especially in Angara land, collections are quite abundant and have many specimens and sometimes mixture of species. we will not discuss all the record. It is quite tens and hundreds names with very difficult to provide in short time. But we will speak something about mosses which are especially abundant, especially important to say something if they relate to extant mosses or not. And tremendous step forward for in studying Paleozoic mosses have been done by Maria Friedrichovna Neiburg, who described 9 genera from 15 from
just click once again.
Here, or is it okay?
No, you left the full screen mode. Would you return to it?
Shall I? It is no. It's
sorry, Jane, could you help?
No, I can start only with beginning. So
okay, and list till the slide you need.
Yes. It is not seen all the screen for me. Neiburg described, among others, Protosphagnalis, genus Protospagnum, and erected on it the order Protosphagnalis. What is this Protospagnum? In habit, it much more similar to what we know in extant moss flora as Mniaceae. Size, leaf shape, and even style of variation of leaf shape is okay. And this Protospagnum is similar to sphagnum in cell areolation, but it have nothing to do in general appearance, as you can see here. Neiburg referred to Protosphagnalis 3 genera were shown here. They differ in some in number of cells per loop among darker cells, and she also described 6 genera which she referred to Brialis. Brialis that time was understood quite broadly as all mosses except Sphagnalis and Andrealis. So that's quite inexact placement, but in some mosses that she referred to Brialis, there are some differentiation somewhat similar to what we have in Protospagnalian mosses. And further studies reveal even more gradual transition from protosphagnioid and bryoid areolation pattern, so it became very difficult to decide about a specimen where to place it if you need an identification. Moreover, some specimens have areolation of one order Protosphagnalis at the leaf base, but in distal part of leaf you see it bryoid areolation. One example, another example, and such examples are more and more were found when we got abundant collection of from Pechora coal basin. And another thing which is important for our study, that we found that unlike extant mosses, the cell divisions are terminated not at leaf base, but often in the middle or at near the apex. And this is interesting that above these small cells, differentiation goes to the protosfagnalian pattern, like here, and closer to bottom, they are more similar to genus Intia. And we thought how to do analysis of this situation better, and developed a special coding of number of cell angles with colors. It looks maybe very strange and difficult, but it's not really difficult. You see, originally cells are in fast dividing area are approaching to quadrate shape, and then after a certain shifting, areolation is developing to hexagonal pattern, and most mosses in extant flora have hexagonal pattern. But if divisions are going in a different way, we may see here so-called tetrads, T-tetrads, like in modern sphagnum. And here is something which is happen when divisions are going further, and here is one T, here is another T, and it's already protosfagnalian pattern. I'll show with some examples how it works. For example, extant mosses usually like these hexagonal cells forming such antagonetic cell packets throughout the leaf. In sphagnum, first divisions are equal, but unequal or asymmetric divisions are started very close to the apical part of leaf, since fourth or fifth division, and these divisions produce pentagonal cells. And after second division, so forming T pattern, we can see here 8, 5, and 5. So altogether is 18, dividing in 3, it's 6. So it again appears like overall it's kind of hexagonal cells, but it's no hexagonal cells except some marginal parts. And when we study protosfagnalian moss, we may find some leaves that are full of hexagonal cells, but in other hexagonal cells are almost absent. And how this can be shown in examples with a small cell in the middle of leaf. On the left are shown cells of typical for genera described by Neighbor, and we see here Intia pattern at the bottom. Here the variable in the central part, and then here we see protosphagnoid areolation without hexagonal cells. So once again, look at this scheme. So this transitional pattern appears with two rows of cells where one row divided and another row is not divided. This it can be called ready steady, but it can go both to hexagonal cells and protosphagoid cells. Let's see again. So this pattern can divide to one way or another. And then we look at more protosphagoid cells. We see that it's a fantastical plastic changes which can involve the whole leaf lamina or be at places or sometimes be just in one row among hexagonal cell pattern, which means that these changes within protosphagnalia group are very easy, and they are always happen. And in most mosses, you can find both areolation pattern. And after that, we think that the definition of neighbor that protosphagnalian mosses have to have clear differentiation, darker narrow cells and lighter, more isodiametric cells. We now think that the presence of these T tetrads, T triads, and TT triads is much more important for definition of this group of protosphagnalia mosses. But if we assume this definition, what's happened next? Sergei Viktorovich Meyen described the genus Rhizinigerites because it has very abundant rhizoids on stems. This is very interesting moss, which have also rhizoid pores called so because parallel to rhizophores of lycopods, completely leafless axis with rhizoid bundles and sometimes broken. And now you see that rhizoids are not near leaves, which is quite a strange situation. And in extant mosses, probably only Takakia, one of the earliest divergent lineage, have something like that. But if we look more carefully in additional material of Rhizinigerites, we see that it time by time have this protosphagoid areolation pattern. So it probably have to refer to protosphagnalis, despite it have no border and have some differences in leaf shape, and also the lamina have many holes. Some cells are falling off, and it's really difficult to say if it is an artifact of fossilization or it sometimes happen when these moths have been alive. But also, this Rhizinigerites at base have very strange veins or lateral branches of costa, but I prefer the term veins because it is very similar to what Neighbor call net venation in the genus Polycyvia. Paleozoic have not so distinct T pattern, but it has this kind of veins, lateral and also sometimes anastomosing. Actually, it's very difficult to decide that this is really moss, because originally this plant had been described as gymnosperm, valse, and a neighbor actually she used Dara film technique, putting nitrocellulose on a rock and then put this nitrocellulose with some cells in slides. So the general appearance of shoots is better in this approach, but cells are not so well represented as in if they being solved in fluoric acid. And such lateral veins were also noticed in two other genera by neighbor in Bandave and Uskatia. So it also link with mosses to Protosfagnalian group, maybe. Further on, look at this moss. These have nothing similar with what we saw before as Protosfagnalian mosses. It was described from Aristova place with in Vologda province with quite rich moss flora in the late Permanent. 13 moss genera have been recorded from this area, probably the largest number. And many mosses from this Aristov have been compared with extant groups, for example, with Physidens, because of very peculiar prominent border. Despite there are some heterogeneity in cells, but a new collection from Aristov showed that these mosses referred to the genus Cervictia have sometimes perfectly Protosfagnalian areolation. So they, by upper part of this leaf, it is everyone would compare it with extant Acrocarpus moss, but it is obviously not so.
Just click once again and it will disappear. Oh, yeah.
Oh no.
No.
Sorry.
Well, some technical difficulties. Sorry, let's wait a bit.
Думаю, лучше закрыть.
Да. Станиславович, лучше закрыть и перезапустить.
Прикольно. Хочешь покажу?
Большая презентация, the big presentation.
Too heavy, perhaps.
How about now? I think it works now.
Yeah,
well, so this is Protosfagnalian, despite by upper part of this leaf, nobody would say this. And there are actually several other genera. I will not speak about all of them. For example, with Paleosphagnum, with big loops, with many cells which are surrounded by small cells, like in, for example, sphagnum. Looking at this Aristomosis, we see that Protospagnalian group enriched a lot, including more and more species which have not been even thought that they can be one day be moved to Protospagnalian. So the puzzle is very incomplete. We are trying to combine all this data for Protospagnalian, and several characters are unique or subunique for this group, and 11 genera now can be placed in Protospagnalian, but also there are some genera which we put in Protospagnalian with some hesitation. I show just one, Gamankovia. It has branched costa. Nothing similar is in extant moss flora. So, but only one character share with other Protospagnalian, so I'm not sure, but maybe further data will show that it is also somehow related to Protospagnalian. But what else we can say about Protospagnalian? We still not mentioned all strange character which might relate to them. Once we described a genus called Bulbasphagnum. It is spheric bodies with rhizoids or very short, something which, if it elongated, you see oblique cell walls, which means that this is rhizoids. And sometimes, quite rarely, we see that these bodies produce stems, and on some stems we see leaves with Protospagnalian areolation pattern. Such bodies probably originated on stem and on costa sometimes. Strange thing. Another strange thing is that leaves are attached to stem by very narrow line. Originally, it was even thought that some small leaves have stalks, so they are petiolate. That's not true, probably because this is very obscure area in the specimen. Probably it's just an artifact of fertilization. But anyway, in many leaves we clearly see that costa and only a little lamina are attached to stem, which is not common in extant groups. And actually what I know is where leaf base is so narrow is only sphagnum. There are some other cases where the leaves looks that they are narrow at base, but careful study show that they are quite widely overlapping, like here. So you see here examples of protosphagnoid mosses, and even there are line of cells in between leaves, which is strange, because if the leaf leaves appear like here, they have no chance to have something in between leaves. Sometimes such thing happens in hepatics, but not in mosses. Also, even in Protospagnum itself and India, costa is branching, and the sides of costa looks unistratose, and sometimes actually neighbor wrote that costa is multistratose or sometimes unistratose. Here we may admit that costa is unistratose, but they took several specimens where costa dark, so looks multistratose, but it turned out that it is almost throughout unistratose. And this is especially strange because Protospagnalian mosses were large plants. Protospagnum itself have leaves over 10 millimeter, and genus Varcotonularia probably has the largest leaf ever found in mosses. Neighbor reported one leaf 16 to 8 millimeter. Of course, there are some longer leaves, but if leaves are long, like in, for example, in Dawsonia, they are not so broad, and the area is smaller. And here you can see another example of this unistratose costa. And looking at these different specimens of Protosognalis, it's very difficult to avoid imagination that these long scales that are spreading from costa to lamina is something conductive. But if they are conductive, what for they can conduct? It's very strange, because mosses are not so big plants, and they are not usually stored anything in roots. They have no roots, but they may store something in these tubers, which actually quite abundant. And in some collection there are tens and hundreds of these tubers, and they are quite hard, have hard epidermis, so they are adjusted to long preservation in soil. So they are not like propagula in many of extant mosses, which are which propagate shortly after they fell off the maternal plant. After those several very strange characters, it is what's the general perception of these Protospagnalian moths? They grow in very northern part that time, and they live close to the pole. It was Cardiatis taiga formed by trees that have annual rings, so it was a seasonal climate. And some of Cardiatis, as some scientists thought, are fast growing. So for Protosphagnalia, the combination of very thin cell walls, unistratos costa, very big size, kind of vegetative, abundant vegetative reproduction. It led me to admitting that they resisted, so survived unfavored time in these tubers, and then grow fast and having need to grow fast, they build their body not that properly as extant mosses they have, which usually made costa with special conducting system. So this is probably a completely stupid idea, but it may be something which allow to combine strange features which we observe in this Protosphagnalia, Intia, and similar mosses Cassiunia. And of course, the growth by apical part of leaf, not only by lower part of leaf as in most mosses nowadays, is possible, is one of possible way to grow as big as they are in Paleozoic. So that's enough for Protosphagnalia mosses, and it's still not add very much to the understanding, but by additional facts we probably finally can come to the understanding what they are and how they live and why they were so different and so unusual in moss world. So not Protosphagnalia mosses are shown here. They also derived from Aristova place, and they have very long acumen. Sometimes not so, but they are very dark and thick wall cells, make them sometimes almost intransparent. When they have been described, they first have been referred to several possible groups among extant mosses: Potiaceae, Seligeriaceae, Dicranaceae, and also Andreaceae. And recently we found better material, and this is very good case when we can more or less certainly be sure that this moss can be related to one of extant group, and Andreopsida. Andreopsida or Andreobryopsida, it's a little bit complicated special story, but you see here the arrelation pattern which never occur in any other group. There are two rows of cells filling subapical part, and apical part is built of or unicellular role. One another Permian moss have been described from Russian Far East, not far from Vladivostok. Actually, now it's Vladivostok area in earlier Permian. It looks very similar to some Polytrichaceae because of terminal rosettes, which are usually surrounded antheridia. By this reason, some publication cited it as a member of Polytrichaceae, but we describe it as Paleocampylopus, which is another group. It belonged to Dicranidae subclass, and now it is in Campylopus belong to Leucobriidae, Leucobriales order. They also have similar densely arranged leaves around antheridia and may look also similar to this. So how to decide about this? If it better to refer to Polytrichum or to Campylopus? Unfortunately, no way, because despite they similar, they have no structure of costa known, and by this reason we cannot decide. This, moreover, with specific structure of costa may appear quite later, much later than the group have been separated from the main stem of mosses in evolution. In flowering plants, it was analysis which show this the similar situation, and they decide they define the stem age and crown age. The stem age is a age when the extant clade separated from sister clade of gymnosperm, and this age can be quite late, quite old, but the crown clade, which combine taxa of only extant groups, it is the distance is 100 million and half. So unfortunately, leaf costa in mosses, it have been studied in Paleozoic, one Paleozoic specimen and one Jurassic, and both have undifferentiated costa. So complex structure of costa, surprisingly, it appeared in moss evolution only in Cretaceous, or we simply cannot find complex costa in earlier deposits. So we actually need more studies of plants that are looking like these, because terrestrial plants probably are much more informative for moss evolution not aquatic plants as were probably protosfagnalian, at least protosfagnum itself, and closely related to it and similar looking plants that quite probably grow in shallow water. At least we see in collections with protosfagnalian moss almost always numerous oogonia of Horatzian algae. So, but what we can say definitely is that this moss, despite we don't know if it is polytrichoid or dicranoid, we can say that this is not protosfagnalian, and so we have at least some non-protosfagnalian groups in Upper Paleozoic, but much less than we thought earlier, because in publication of 1990, most genera described from Aristova were referred to, or not referred, but compared in morphological character with this or that extant moss group. So extant mosses are very extant moss group are almost not represented in Paleozoic, and quite many protosfagnalian appeared, and maybe even some more will appear in the future. So this is my view on the situation now. So in short, in brief, conclusion is that Permian mosses were diverse and locally abundant, that protosfagnalian are much more represented, and we just under-recognized them in fossil collection. But anyway, some extant group are represented in late Permian, and the leaf structure was completely unusual for more for extant mosses, and probably it have many function way to function to so that is not similar to what extant mosses can do. So finally, I must say great thanks to all my collaborators who helped me to collect material and study it, prepare slides, and study under special microscopic technique. And also thanks for attention, all of you, and all who will be interested to see this record after that. So thank you very much.
Thank you, Mikhail, for the beautifully illustrated and very informative lecture. Thank you all for listening it. Thank you. Well, now, please questions to Mikhail if you like. Raise your hand or write your questions in the chat, or raise the hand, the virtual hand, and ask Mikhail about mosses. Well, and you may switch on your phones. Sorry, no discussion so far.
Here's one question about review paleobotany palynology. Probably I didn't know that yet.
Mikhail, I see your hand, but the phone is switched off. Could you switch on your phone? Okay, you're welcome.
Yes. Hi, nice talk, Misha. I was curious if there's any, or I may have missed it. Are there any living mosses where the leaves develop um, apically for a longer period of time?
Sorry,
At the tips,
what?
like you showed in the Protosphagnales, if there are any living mosses where leaves develop like in the Protosphagnales with a more extended growth period at the tip.
Есть ли живые мхи, у которых такой же, как у протосфагновых, развитие листа?
No, I don't know. It could be some small teeth which may extend at different part of leaf margin, but not similar to what we have in Protosfagnalian. This is completely unique both in the middle and in the article part, I think.
So I actually so in all or most living mosses, leaves mature from the tip toward the base?
Here is very difficult to define actually, because at least in all mosses you would find small cells in the basal part if you see very young leaves. Even in sphagnum and further on throughout the phylogenetic tree you would find this pattern. It had been described in the beginning of 20th century. It was big publication of Patier about that, and yeah.
Okay, thank you.
It's difficult to believe, and many physiologists disagree, starting to look and all see the same.
Thank you, Mikhail. Any other questions? And you may pose it in Russian if you prefer.
Можно я по-русски сформулирую?
Welcome.
На некоторых слайдах, которые ты показывал в самом начале, я не помню, протосфагнум это был или что-то типа протосфагнума, были клетки такие большие и изометричные, но более темные, чем все остальные. Это какая-нибудь, так сказать, специфическая черта или вот, ну, как это можно объяснить, что это такое?
I'll translate. The question is that in many protosfagnalian mosses we see a little bit larger cells which are darker in color, and they are evenly or unevenly spread throughout the leaves. It is not always, but quite often. I would thought about fungi and something like that, and it can happen at the stage of fossilization or decaying of them. But really, I don't know. We will try to see what we can find. Sometimes they have also some crystal of pyrit. But that is not obligatory associated with darker cells. We are planning to study. В общем, я не знаю, Леш. Мы старались понять, но не смогли разглядеть ничего такого, о чем можно было бы сказать.
Спасибо.
Thank you. More questions? Well, I do not see. Oh, one more from Mikhail, and you may switch again your phone.
Yes. So in the slide where you showed Protosphagnum myani,
Мгм.
there was a leaf that had an interesting cell pattern. If you could go back to that slide.
М, Just a moment, I'll
Just a
try to,
moment.
if I can, yes, I'll manage, I think. Um,
Yeah, keep going.
This one, Paleosphagnum maini. You mean
Yes.
this one?
Yeah, I think it's this one. If you can go another slide back.
No, another slide is it's already
okay, yeah, I know
another
that, yeah.
genus.
Yeah, okay. So it's
So
that one.
it's
Yes.
only one slide. But
Yeah.
I did say everything about all mosses. Interestingly, Cervictia, Gamankovi, and with Paleosphagnum, we managed to obtain costa cross section. It is not unistratose. It is multistratose. So unistratose part pertain mostly to Protosphagnum group, Rhizinigirites group, and Intia group.
So my question was about figure B, where on the right-hand side, the leaf margin has a little sinus, and then the cell patterns seem to converge toward it. Is that some kind of response to wounding, you think?
Just a moment, because my in my screen I cannot see. B is the second from
that one. Yes.
This one.
Yeah, so on the right-hand side, the leaf margin has a little indentation.
I'm not sure that this is a panel about it.
Yeah, right there, a little bit lower. There, okay,
this one?
yeah, around the lower, even lower. There and below that.
It is covered by.
Yes, see how the leaf margin is like that?
Yeah, it is. I think it's kind of artifact of fossilization.
Distortion.
You think so? But look at the cell patterns. The cell patterns converge toward that area. So it means that they develop like that.
Yes.
Yeah, there.
Yes, I understand it, but it probably have been eaten by somebody. Or...
Okay, yeah. All right, that's what I thought. All right, thank you. So that's a direct developmental response to wounding.
Yeah,
of
it
the
is
living plant.
quite likely.
Yeah, I just wanted to confirm that you think so also.
Thank you. Well, more questions to Mikhail. I do not see any more hands. Perhaps we have done with questions. Well, if so, thank you everybody for coming, and thank you, Mikhail, for giving us such an interesting and beautifully illustrated lecture. Thank you, and I hope you see you all next time. Thank you.
Thank you very much.
Thank you very much. It was very
Thanks.
interesting.
Bye-bye.
Bye.
Thank you.
Bye.